Imagine a world where it is possible to create an exact genetic copy of yourself. The idea has fueled science fiction for decades, but what is the reality of human cloning today? While many international agreements and national laws prohibit reproductive human cloning, there is not one single, universally binding global law. However, the overwhelming global consensus is against it. This widespread rejection stems from profound ethical concerns. One primary concern revolves around the moral status of the embryo, debating whether a cloned embryo holds the same moral value as a naturally conceived one. There are also deep worries about human dignity, fearing that cloning could commodify human life or diminish the uniqueness of individuals. Critics also highlight potential risks to the cloned individual, including unknown health consequences and psychological issues. Furthermore, the specter of exploitation, especially concerning women who might be used for egg donation or gestational surrogacy in a cloning process, raises serious ethical red flags. These complex issues have driven nations and international bodies to take a strong stance. So, how does this translate into action? Many countries have enacted specific laws banning reproductive human cloning. International declarations, such as the United Nations Declaration on Human Cloning, recommend that member states prohibit all forms of human cloning incompatible with human dignity. For researchers, this means adhering to strict ethical guidelines and national regulations, focusing instead on areas like therapeutic cloning, which involves creating cloned embryos for stem cell research, not for gestation. This distinction is crucial for understanding the current landscape of biotechnology. It is important to differentiate between reproductive cloning, which aims to create a full human being, and therapeutic cloning. Therapeutic cloning, often utilizing a technique called somatic cell nuclear transfer, or SCNT, involves transferring the nucleus of a somatic cell into an enucleated egg cell. The resulting embryo is then used to derive embryonic stem cells for research into diseases, without the intention of implanting it into a uterus. This distinction is vital in ethical discussions, as therapeutic cloning is viewed differently by some, though it also raises its own set of ethical considerations regarding embryo destruction. The global ethical landscape surrounding reproductive human cloning reflects a powerful collective agreement on fundamental human values and the protection of human dignity. While specific legal frameworks may vary, the core principles of preventing the commodification of life and safeguarding individual uniqueness remains paramount. This ongoing dialogue ensures that advancements in biotechnology are pursued with careful consideration for humanity's profound moral responsibilities, guiding research towards applications that genuinely benefit society without compromising our shared ethical principles.